Welcome back. This is lecture 7 in computational bioengineering, dynamics on networks. Today we have a bunch of things to cover. We're going to talk a little bit about some things we tried last week that didn't work exactly the way we hoped they would. And we're going to have some student ideas about projects, and then we'll see how far we get moving forward with the idea of analyzing dynamical systems. So we're going to start out by talking about running Tellurium in Colab. We will also use the MakeSBML function that we didn't get to work last week. And then I'll talk a little bit about using variable stoichiometry in Antimony. And then we'll have the student project presentations, and we'll talk about what kinds of things you can do with networks and how to ask questions about them. And then we'll see after that how far we get going forward. We'll talk, I hope, about Antimony events, which are a simple tool in Antimony that's quite useful when things happen discretely rather than as dynamics. And then we'll see whether we're able to get to some motif analysis as well. So one of the things that we did, and perhaps Hayden can put the link in the chat, was it seems that, of course, this class covers many different topics and many different ideas, and some of these will be unfamiliar to different people. And so it would be very helpful if you would tell us when there's a term, a concept, a method that is unfamiliar, that needs to be defined. Maybe we talked about it in class, but it wasn't clear. Could be something that you run into in the lecture, in exercises, in the homework, or while you're reading. And we'll do our best to come back, and when people seem to think that there's something that needs to be clarified, we'll try to offer in-class definitions, clarifications, or explanation of these concepts. We've created a Google Doc for that, and if you could post your comments anonymously, and then if you think somebody has listed an item that's important that you'd like to see, you could put an extra star or something in front of it so that we know, gee, this one is, we'll try to cover all of them, but if there are a lot, we'll focus on doing the ones that have the highest priority first, which are ones that multiple people find unclear. Okay. So I wanted to talk about Colab. I know a few people are using Colab already. We've had some issues with Tellurium on NanoHub. In particular, the Tellurium on NanoHub is an older release, and it also has limited availability of IPy widgets for doing user interface, because NanoHub only supports a subset of IPy widgets. And it also is not so easy to upload files to NanoHub when you want to distribute your code. And so we've thought about for some time the possibility of switching over to Colab. Since I was used to teaching out of NanoHub and all the exercises I had were NanoHub, I was a little bit reluctant to do that, but I bit the bullet and was talking to Herbert and Lucian, who maintained Tellurium, and they suggested that it's not so bad to use Antimony and Tellurium in Colab. Now, if you look online, you need to, I'd like people to do this. You need to go to Colab. Anybody with a Google account has a Colab account already built in. Depending on exactly how your computer is configured, it may have to do a download to your local computer or not. Once you've opened Colab, and it's one of the many options in the list of things that Google offers you once you're logged into Google, you have to create a new notebook, rename the notebook. You'll need to install Tellurium. And if you look online, the instructions will be pip install -q Tellurium. And then you should add a cell and type import Tellurium as TE. And then you should be able to use Tellurium and Antibody as usual. And there's this save option at the top right, which will let you publish either to GitHub or to a Google Doc, a Google Drive. Now, those instructions don't work on my computer. I know some people it'll work on, some people it won't. And the issue is depending on what other Python packages you have locally loaded on your computer. And so if you don't have any other Python locally loaded, this sequence should work. But if it has a problem, then you're going to need to do two additional things. You'll have to do !apt-get install libncurses5, and then an exclamation point before the pip install. And the one annoyance factor here is that when you run this notebook, the first time you run it, it will crash. And you will then have to go under runtime and do a restart runtime and do it a second time. And I do not believe it's possible, and maybe somebody can help me with this. I don't know whether it is possible within the notebook to tell the notebook to restart. In other words, I don't know whether there is a command, inline command you can use within a notebook to tell it to restart. I was going to look that up, and I didn't have a chance to look it up today. But this should work. In the PowerPoint, I have a link to a Colab notebook that has the things that you need. And let me put that link in the chat for you. If we're going to publish our projects, we have to have a way to do it. If we're not going to publish them through NanoHub, the other option is Colab. And as I mentioned, Colab has a lot more flexibility in terms of what packages you load and how you distribute it.
Actually, I know that you had the DOIs in NanoHub, and you can do the same when you have a GitHub, and then you have like 10 auto, you can get a DOI if this is something that people would like to have.
Right, then it's a little more persistent, so it looks more like a publication for your records. That was why I was a little disappointed that you might have to do a restart to run it, because ideally you want a single button execution. But I guess if the notebook says, if it doesn't run, please hit restart and run it again, it's not the end of the world. So if people are familiar with how to publish using Colab, I'd very much appreciate your guidance about the best way to do that. We'll try that out this semester. I think you will find that it's less work. In other words, creating the GitHub repository and then uploading it to NanoHub is a little bit of a chore. It's not terrible, but it's a little bit of a chore. With Colab, you could publish it through Git, or you could simply publish it through a Google Drive. And so there are a variety of options here. Again, I think NanoHub has a lot of great features, but it's quite old. It was developed long before these other server methods and sharing methods were developed. And it was developed at Purdue. And of course, Colab has the might of Google behind it, and so it's more polished. Okay, so by all means, let me know what you think. I've been using it and finding it quite convenient once you get past this business of having to do the restart. So the other thing that I wanted to come to is downloading SBML models for BioModels repository, converting them to Antimony. And as we found out last week, the BioModels repository seemed to be having some glitch where when you tried to view the models, it wasn't working, and when you tried to download them, it wasn't working. And that is, as of yesterday, was still an issue. However, and I have the slides from last week that are included in this lecture, but there is an alternative, and I would like people to try this alternative. And again, if Hayden, if you're there, maybe you could paste this link into the chat for me while we're talking. So Herbert Sauro has created a very nice GUI interface that will do the model translation. And it's called MakeSBML. It uses an API for BioModels that didn't break. And so it will work. And so I would like everybody, this is now a little exercise, I would like everybody to navigate to sysbio.github.io, MakeSBML, and let me pull that here for you. So I'll give people a minute. Please try that and make sure you get there. Give me a thumbs up in the chat okay, great. So that worked. So once you're here, the steps are going to be navigate to that. We'll first repeat the exercise we did before. We'll search for EGFR. We'll find the Bianconi 2012 paper. We'll download it. We'll translate it. We'll copy it into a clipboard, and we'll paste it into a Tellurium execution model. We could use the one that I shared with you in Colab, which is how I've been doing it, or you could use whatever your local Tellurium is, okay? And I'd like everybody to do this. I'll walk you through the steps. So the first thing you're going to do is in this little search bar, type EGFR and hit return. Hit download here. So it should give you a list. So everybody got that? And you should see a long list of possible projects. The second one that I have when I pull it up is Bianconi 2012. So click on Bianconi 2012. And that will replace EGFR with BioMD 0000427, which is the UUID of that particular model. And then again, hit download. So that worked for people? Okay. And so what you should see is a bunch of SBML code here on the left. And it probably will automatically translate it into Antimony. If it doesn't, hit the little translate button here in the middle. That will take you back and forth between Antimony and Tellurium. And now at the bottom, you have options: save Antimony to file or copy Antimony to clipboard. So hit copy Antimony to clipboard. And then open your Tellurium. Start out with model string equals triple quotes. Paste what you've just written into that string. And then do a TE load A, r.simulate, and r.plot, and see what happens. Okay. So copy the code. Here in the code that I shared with you in Colab, import Tellurium as TE, import NumPy as MP, model equals triple quotes, paste it there, r equals TE load a model, r equals r.simulate 0 to 10, r.plot. And tell me what you get. And I'm not saying it, you don't have to do it in Colab. If you have a local install of Tellurium, and you'd rather do it there, that's fine. If you're using NanoHub Tellurium, that's fine too. The only thing that would be different in Colab is that you have these 2 lines of code to install Tellurium at the beginning. but you might not have in your other installs. Try restart run all.
Oh yeah, now it works. Thank you. Yeah, I made them.
I don't understand why you have to restart it, but that seems to be the
Most probably, what I guess, because you install with opt, you install Linux libraries, and most probably it had them not before in the path, and when you reload it, then you have them there.
I think
What I guess.
that's probably correct. It's as I say, it seems funny because if the install works the first time, you don't know why should you have to do it twice, but that seems to be how it works. It could work if you do an import, if you do under runtime, if you go under runtime and hit restart run all, it should work. Again, I'm basing it on my experience using it.
So maybe I said it short again. So what you have to do, what I figured out so far, is you do opt install, then you do the pip install, and after that, when you would run, then it crashes, but then you restart, and then you don't do the installation anymore. You just do the import from Tellurium or TE, and you run your code. But the installation, somehow you have to restart after installations. So yeah.
For the result of what we just plotted, that should be the same thing as the one notebook that you already shared with us.
Did I share the notebook which already had the code that you were downloading and pasted in?
I mean, maybe it's just a coincidence, but I get the same. Maybe it's not the same, but it looks almost identical. I was here, I copied this to clipboard, click this.
Yeah.
Went over here. I guess I could just do it now. Just
That's
copy
it. That's the whole
Copy.
thing.
Okay. Perfect. I guess that's the same thing.
Yes, nice.
Yeah, that's it.
Okay, cool.
So that makes it pretty easy to download and run a model from BioModels. That's what I had hoped we could do last week, but with that download button and BioModels not working, it was a problem. But the way we were doing it last week, we had command line interface to do the translation. Now it's a GUI interface.
For the do you know about the search button? Is it BioMod? Is it, how do we cite? Oh, I guess you can see it in the SBML.
It should be in the SBML, but it's also if you get the you see the ID number.
Yeah.
So the ID number, if you search that ID
Search it
number,
up. Okay.
it will pull up the reference.
Okay.
That's a unique locator for that model.
I think it's even in the code, in the Tellurium code, the link.
Tellurium, the Antimony,
Actually,
SBML normally suggests that people document their models.
Yeah.
And models that are annotated and uploaded to BioModels are supposed to have a certain minimal amount of annotation in terms of authors, sources, and so on. But it's not always going to be true that every model you run into that you want to translate will be annotated. And so, William, your point that you should look up where it came from and have a proper citation is a good one. So one thing that I find a little bit confusing about Colab is Colab tends to overwrite your existing files. It doesn't like to rename things very much. It likes to auto save things on top of what you did without your hitting save. So I do recommend saving a copy or renaming things pretty frequently when you're using the Colab. Again, I'm not a Colab expert, so you probably have as much or more experience than I do with it. And so that's what I got. And I'm not saying that this is the most beautiful thing in the world, but it's a complex model. We were able to download it and run it. And once we got Colab working, we were able to download it and run it in a minute or two. That's not bad for model replication. If you try to replicate a typical published computer model, it takes hours, days, or even weeks sometimes to try to replicate it. And so this really is quite a nice feature to be able to download and run models. Okay, so my next little exercise, and again, I don't want to take too much time, but I think it's worth it, is why don't you try doing the exercise I hope people would do, which is go back to the MakeSBML, search for something that you're interested in, and do the same thing again for a different model, not the EGFR model. And then maybe one or two of you could tell me what you got and show me what you got.
I'm getting an error that I'm trying to figure out what's wrong. It's not working. So
okay, do you want to screen share it and we can look at it together? I can't necessarily, it's possible the model doesn't run in Antimony. It's not every SBML model will.
So this is a similar model to what I'm doing now, or what my project is on. But we have okay, I just want to see the error message. Line 118, syntax error. Oh, end. Should I just delete end?
No, that usually,
I thought you need end.
unfortunately, that usually means that there is a typo in the model.
Okay.
And that's sort of an annoyance.
Well, no, yeah, I don't know. I don't understand why some of these have double. I don't know. I made like, I have no idea. Okay.
So
Huh.
it's possible that this is actually a translation error.
Yeah.
Or that
That's all fine.
or that there was a variable name that was illegal in Antimony that was legal in SBML.
Okay, wait, maybe if I changed something. Wait. Let me just try it one more time. Here. Okay. Yeah, this is what happened. It was weird because this was going on. So
Giving a sorry. Let me see. I'm not sure.
oh, well, I mean, it's just like it's not the triple quotes. It's thinking that the triple quotes are here, but they're not. And I think that's what is messing it up. KS2__ is K. Like, do we even need this? These are just display names. Does it really matter?
I think if you got rid of the display names, you'd probably might fix it.
Yeah, I mean, I don't think we need that.
So basically what's happened here is that there's something that's a variable name in the SBML.
Yeah.
Is a reserved word in Antimony.
Yeah. Okay.
And so that's
unknown model qual.
okay, well that one maybe is something.
This, I think you just delete this.
Yeah, you don't need to name the model.
Right. Still an error. Unexpected end. Huh.
Again, that means that there's still it's probably, I apologize. This is one of these things. Automated translation is not always trivial.
Got it. Okay.
So almost certainly what has happened here is that there is still something like somebody's used a character that Antimony thinks is a reserved character.
Got it.
For example, as you pointed out, a triple quote would be read as a string character when it's probably trying to represent the time derivative.
Yeah.
In this case, unfortunately, we'd have to go through and look by hand.
Okay.
And so I'm sorry that you picked one that didn't work.
Yeah.
But I guess it's part of life that these things don't always work. Did
Well,
anybody get it to work?
I picked a multiple myeloma model because I'm interested in multiple myeloma. And oh, that's wrong. So this is what I copied, yeah. So yeah, and I not even studied, but when I run it, it looks like this.
So it's not doing anything.
Yeah, so it's just,
So,
but it works. It doesn't give an error. So would have now to understand what goes on with these different AB.
you might have to
CBT.
increase the time.
yeah, I was going to say, try running it for a longer time.
I think so. That's the.
That won't change the time. That changes the granularity.
Oh yeah. Oh, sorry. Yeah.
The time.
Yeah.
So I was going
Yeah.
to say, William, it's not much fun. One of the problems with using Antimony is because of the way it's parsed. Yeah, no, it's not.
Yeah, I think it's not much going on.
So well, it did something at the beginning. It's not exactly constant at the very beginning, I think.
Yeah, so when I go, but then it should by 100.
Why don't you try one? Try time one.
This one is a little bit curved.
Yeah. So it's
or 110. Yeah.
So there we would probably have to look at there probably are some particular additional values that you have to change to make that model do something interesting.
Yeah, by one it's just yeah. Okay, but it would work from the translation.
But at least it executes as.
Yeah.
I was going to say, William, it's not one of the problems with Antimony is because of the way it's parsed, the error messages are not always informative. So for example, unexpected end usually means that there's a missing parenthesis or a quotation mark or something like that. And the best way I have to do it is something that's rather tedious, but works, which is I go from the bottom, I comment out the entire simulation. And then I just gradually uncomment from the top down until I find the error. And that's not, of course, if it's very long, that's boring, but you can do it in blocks. You can comment a block of code, check that, uncomment it, check it, the next block. And so that's how I look for parse errors and syntax errors. If I have a situation where you run it and it crashes, then there are two steps that I would do. The first one would be essentially what Elmar just did, which is I would run the simulation for a very short amount of time. Or if I suppose I start running from 0 to 100 and it crashes, I'll try for running from 0 to 1. Does it crash? 0 to 0.1, does it crash? Because if it starts execution at all, that tells you something about the way the simulation works. And then you can find the time at which it fails and try to debug from there. If it crashes, even for a very short amount of simulation time, that's almost always a divide by zero error in one of the rate laws. And so if you have a crash immediately on execution, then you look in the rate constants, those things after the semicolons in the reaction declarations, and see if something in the denominator is 0. And that's almost always what that is.
The most probably the problem in your model before was that they used triple quotes, and that's why it thought that the Python string is finished.
Yeah.
And then you loaded not the whole model, you loaded only till these 3 triple quotes came. That was most probably the problem in the old problem that you have, because you cannot, this model is not supposed to have any triple quotes, otherwise it thinks that the Python string ends. And then what you can sometimes change is the double quotes or the single quotes, right? You can use 3 double quotes or 3 single quotes, but I think what I saw on the fly when you had it was that was the case by you, that you had 2 times triple quotes.
I suspect that what they're doing is that the time derivative is sometimes represented with prime. with the prime, which is a single quote.
Mm.
And so probably they were having the value x, the time derivative of x, which they wrote x prime, the second time derivative of x, which they wrote x double prime, the third time derivative of x, which they wrote x triple prime. And in SBML, that's allowed, but Antimony will not allow you to write that.
Got it.
Just the way the variable name gamma is an allowed variable name in SBML, but in Antimony, gamma is reserved for the gamma function. And
Mm.
so just the way you can't name your chemical species time, if you name your chemical species gamma, Antimony will throw a strange error saying, why are you trying to reassign the value of a function?
Mm. Okay.
And so the ability to interconvert between SBML and Antimony is pretty good, but it's not perfect.
Mm.
And so I'm afraid you got hit with the first example you picked was one that it didn't translate.
Okay, so I did the exact same thing. I got it from online, and then run it, and I did what you were saying. Like I adjusted the time blocks to like the perfect, sort of figured it out, and then this is super helpful for what I wanted to do, or informative. So it was good.
So actually, I have a suggestion for that. You see how the simulation is a little bit jagged there. The peak
yeah.
has sort of a sharp point.
Yeah.
Where you say r dot simulate 40, 80, 100.
Yeah.
Try making it 1,000.
1,000.
Yeah.
Oh.
That's the number of little sub-steps. If you have very sharp functions, then they can get a little bit jagged. You see how it's smoothed out the curve there a bit?
I see. Okay.
If you see angles, sort of bends, sharp bends or spikes in your simulation, then you usually have to use a few extra points. You basically have to make the temporal resolution of the simulation a little finer.
Okay.
So I hope this is useful to you, because this kind of debugging discussion, it's hard to have these kind of debugging discussions in the abstract. I think I talked about these things. I showed you a slide about this weeks ago, but it's not going to mean anything to you until you run into the problem.
Yeah.
So then working with you to try to debug in this way, I hope is something everyone can learn from.
Yeah, so this was just an E. coli, like they had several E. coli living in the same species. Some of them were predators and some of them were preys. And this is just their population over time. It's pretty interesting, I think. And if you increase the time, like 400, they go through like periods, which is pretty interesting, I think.
Okay, pretty neat. So you got an oscillatory predator prey. You asked a question about some of these annotations. And again, SBML is big on annotation. Antimony has some support for this. So one question that you have in biology is that terminology is often ambiguous. And you'll say something like cancer or breast cancer. Well, there are many kinds of breast cancer. Or you'll say this is a T cell, but there are many kinds of T cells. And if you want to build computational models which can be interconnected to each other, or you want to build computational models where somebody knows how to change it, it's useful to actually annotate when I say VEGF, I really mean this particular molecule. When I say T cell, I really mean CD8 plus T cell. And so there are ontologies which contain definitions of a variety of terms and concepts. And blurb here in Antimony is essentially saying that you're going to ignore this, you're not going to parse it. If you go back down to the bottom where you were, you see here it says blurb is, so that's the name of the model and the reference, the reference for the model. Blurb, model entity is, and then these are the BioModels identifiers. That's actually that unique identifier for the particular model you're running. But if we go down a little bit lower, PubMed 184144488 is the identifier of the paper in which this model was published. And then the last two, and this one isn't really useful unless we look it up. Go is the gene ontology. And so this is a reference essentially to an ontology, to a dictionary, an accepted international dictionary that defines those terms. And so if you then searched on Go 0009372, it would take you to a definition. Now, it would be more useful if we knew what it was connected to. Here we have, for example, A2, identity, CHEBI 55474. That's saying that this symbol represents this particular chemical species. And so this is a way now to annotate your models so that if they were going to be used, say, in a commercial or regulatory setting, the definition, this is this very specific chemical. It's not just toxicant. It is this particular chemical. And so then you'd want to annotate it with a specific identifier for that specific chemical. Does that make sense? This isn't going to change how the model runs. This would be used as, this is essentially metadata that would help somebody if you published the model and somebody were doing a search in Google or elsewhere, and you wanted this to pop up when they searched that particular concept, this would allow that to happen. Or if somebody were looking at the model and they wanted to know, what did you mean when you said this? Instead of looking at the paper, they can click there and it should pop up the definition. I don't know if they all will. Those Go ones should pop up the definitions. Okay. Any questions about that? Does that make some sense? So this is not, none of this is things that have to do with model execution. They all have to do with annotation and description of the model.
What about this part?
Okay, so compartments we haven't talked about yet. Compartments. So by default, Antimony thinks of all of those chemical reactions occurring in the same place, being mixed together. But for example, if I'm going to write a pharmacokinetic model where I have a model of the lungs and a model of the heart that are connected by blood flow, I could define a box, a compartment that says heart, and then the reactions that occur within that heart compartment, and then a box that says blood, and the reactions that occur within the blood compartment, and then a box that says lungs, and a set of reactions that occur in that compartment. And so compartments are a way to say where things are. And in particular, if I'm talking about amounts of material rather than concentrations, if I go from one compartment to another with different volumes, then the amounts can change. And so in this case, when I define compartments, I've defined a compartment called environment, and I'm saying these variables live in that compartment. In this case, it may be there is only one compartment, but environment here will probably be a volume. It'll be the size of the volume of that compartment. So in that case, if you're interested in that, and if you're going to do PBPK modeling, you'll need that. I recommend that it's discussed in the textbook, and it's also discussed in the online manuals. It's something that I typically don't cover in this class because unless you're doing that kind of modeling, you don't need it for the basics. But it is, I've mentioned, I think, a few times that there are plenty of additional features in Antimony that we're not going to be able to cover in a short 16-week course. So that's one of the ones that we typically don't cover in detail. But it's available to you.
So I tried to grab a model from BioModels, and it gave me a giant string with a lot of stuff.
Yeah,
And I
that's,
don't.
yes, that's typical.
Okay, so do I put this into the load A function like
Yes,
this? Okay.
you need quotation, triple quotes.
Oh yeah. And then just run.
Yep.
Okay. So this is viral infection. Oh, all right. I have to reload. It's reset.
There you go.
Interesting. Infected cells.
It's pretty powerful, this method.
Yeah.
I understand for William, it was frustrating that the first model he picked didn't work. But the fact that you could do this at all is actually to me, is quite impressive.
Yes, it is.
The fact that it doesn't take that much work. And even debugging it, again, in the old days, you would have had to write to people. They would have sent you a cassette tape or something with the model on it. More recently, there would have been supplemental material with 5,000 lines of C++. You would have to figure out how to compile it. And then you'd discover it didn't work on your computer. Or they left something out. The fact that you can download and run these models and convert them between languages, and most of the time it works, to me is very impressive. This is the hard work that Herbert spent basically 25 years of his life doing, is creating these standards so it's possible to do these things. I'm not trying to force you to use Colab versus whichever IDE or whichever Python framework you want, but I did want people to at least try the Colab so that it's available, because it's a way of publishing your results afterwards. And if people have suggestions about the best way to distribute Colab notebooks, how to get a DOI and these kinds of things, and if they want to show the class, I think that would be great. Okay. So the last thing that I wanted to do, and I did create a Colab notebook for this. Remember, there was this question that came up when we were doing Hill functions about variable stoichiometry, changing the number of molecules that are reacting in model definition. And I had mentioned that we'd asked Herbert and his team to add variable stoichiometry to Antimony. And they did do so. The problem is that the version of Tellurium on NanoHub is an old version that doesn't have it. And so I didn't want to teach it if everybody was using NanoHub, because it wasn't fair to have some people in the class be able to do it and some not. So when I've been teaching to NanoHub, I've been teaching to the older version of Tellurium, specifically the ones that would support, to make sure that everything we did in class was supported. Once or twice, there'd be things where I'd do things that were outside of the NanoHub Tellurium, but I try to avoid that so that it's something that everyone can use. And of course, if it was going to be in your project and you were going to upload it, if it doesn't work in the NanoHub Tellurium, it doesn't work. So if we're now using Colab, one thing about Colab is with that pip, you always get the latest version of Tellurium. And so now we can actually declare, if you have a variable name with a space and then the name of the species, Antimony will interpret this as variable stoichiometry. So if I say E plus N space S goes to ES, it will assume that I mean N molecules of S. So this is quite attractive because I can now have, Elmar had this nice text editing framework to do this, but here we can do this within the language confines itself. And so I thought that was very nice. There are, however, a couple of limitations. And my example code here, and I actually have a Tellurium notebook for it, I mean a Colab notebook for it, is here for N is in N pi A range, reset, simulate, and then plot, and these are the curves for different values of N. There are a couple of things that are a little bit strange about it. First, if I said E plus 4S goes to ES, ES goes to E plus 4S, that would be natural. And if I have a forward and a backward reaction, it would be natural to use the same symbol for the number of molecules in the forward and backward direction. If I take 4 molecules of S with what reaction, I'd expect 4 molecules of S coming out. And Antimony won't allow that. I have to have a different symbol in each reaction. So I'd have to say E plus N to NS here, ES goes to E plus N1S, and make N1 and N equal to each other. I can't reuse the name of the symbol for the stoichiometry. And so that's something, that's a peculiarity of the parser, and we have to live with that. The other thing that's a little bit strange is that I cannot say R2 R dot N equals something. I cannot change the stoichiometry from the Python directly, which is a bit inconvenient. However, I can use a function which we haven't talked about yet, but is not so complicated, which is if I say something, and we'll come back to this later in the class. It's a small point, but one that I didn't want to introduce until a little later. If I say H equals 4, then I can overwrite the value of H. R dot H equals N. I'm allowed to overwrite it. But if I change the value, if I use the value of H and do something else with it, it won't be changed. Equal signs in Antimony are only evaluated once when the string is parsed, or when you say reset. They're not evaluated continuously because Antimony is not a procedural language. It's a model specification language, which is somewhat different. It's a declarative language. So this has some odd things about it. For example, the order of the lines in Antimony generally doesn't matter. If I take a procedural language and I change the order in which I have the commands, it won't work. In Antimony, in general, the order of the lines is irrelevant. If I say H equals 4, X equals 4 times H, in Antimony, X will always be 16. Whether I put X equals 4 times H before or after the H equals 4. Clearly, in a procedural language, that wouldn't work. So that tells you it's different. The ability to do a reset the way we do from Python is also something you couldn't do in a procedural language. However, that also means that if I set N here in this declaration and then change the value of N outside, it doesn't change N in that declaration. There is a workaround for this. Colon equals, instead of regular equals, tells Antimony that it must continually reevaluate that equal sign. A regular equals is only evaluated at parse or reset. Colon equals is like a function. It is evaluated always. It's reevaluated anytime H changes, M will change. and I'm allowed to cheat, I can make n here in this declaration equal to something. And then if I change the thing on the right-hand side, it will force a reevaluation of n. That actually shouldn't work in my view, but it does. And so it means that we can, in fact, change the values of the stoichiometry from the Python, but we have to do one extra step to make it work. And in fact, it wasn't clear that this would work. When I talked to the people at Washington about Tellurium, they weren't sure it would work. We tried it, it works. So you can, in fact, change the values of your stoichiometry from Python, but you have to do one extra step. The fact that there are multiple kinds of equal signs shouldn't be totally alien to people. As an example, in Python, we have two equal signs. We have one equal sign for assignment, and we have one equal sign for a logical equality, check if things are equal. We have the double equal sign in an if statement, and we have the single equal sign for assignment. And so Antimony has an extra equal sign, which is the equal sign that says do continual reassignment rather than parse time reassignment. Again, the thing you have to watch out for is that you can't have the same symbol used twice. So if it's n in J1, it has to be m in J2 and something else in J3. You can't reuse the symbol. And then if you want to change the value, you have to use this equals and then the thing you want to assign it to. Once you know those few rules, it's pretty straightforward. So I think with that, it's time. Elmar and William, I think you said that you were ready to give us a little bit of a discussion of your ideas. Doesn't have to be long. 5 minutes is fine. Try not to make it half an hour. Notice this is the reason I'm having these repeated presentations about your project ideas is so that people can go through it relatively quickly, think about it together, get comments from the audience. It's not at the end of the semester you can do a full presentation. Today it's meant to be something fairly short and informal. William, do you want to tell us what you're thinking about for your project?
What I'm thinking about doing is analyzing the role of a protein called P53 and its role in repairing DNA damage, or its role in sort of deciding what to do when DNA is damaged. So some biological background, P53 is a protein that helps suppress tumors, and MDM2 is another protein that suppresses P53 production, but they have a cyclical sort of positive negative feedback loop relationship in the fact that when P53 increases, MDM2, like the production of MDM2 also increases. And so then the increase in production of MDM2 decreases the production in P53. So it's sort of like this cyclic relationship that keeps everything at a steady state within a cell, that keeps the levels in a steady state. But when there's DNA damage from external sources, like in this research, it was gamma radiation. P53 accumulates because MDM2, the levels of MDM2 drops off and a little bit. So P53 accumulates, and that is necessary to help fix the damage or kill the cell. And from the papers that I've read, and one of them was talking about how this like occurrence, which I can maybe show on this page. So this is this graph of maybe I'm trying to think about the best way to describe this. So I guess, yeah, I should explain this graph. So this is a simple model that I copied from a research paper. It's showing what happens when DNA damage occurs in a P53 MDM2 like system. So around time equals 50, DNA damage occurs, and this results in a quick drop in the MDM2 protein in the nucleus, which results in an increase. I don't know if you can see my cursor, but an increase in P53. And so while P53 is increasing, since P53 is increasing, this causes MDM2 to shoot up because we need MDM2 to keep P53 at a low level. P53's level in steady state is normally super low. So yeah, so if the DNA damage is a lot, or if there's a lot of DNA damage, I don't really know how to gauge that yet, but this would happen in oscillations. So this up and down cycle would happen multiple times. And these researchers were just looking at the frequency of oscillations, and they were investigating the function of the oscillations, because some of them had a pattern, but some were sort of randomized, and it was interesting. Some of these bullet points are just, I linked the translator that we already talked about. The default parameters, it's sort of a simple model, at least like looking at it right now. There are 3 variables that we really care about. But in the back end, or in the actual model, it's a ton of other variables impacting these 3. And yeah, maybe, here's another figure from the paper. This was just showing here is the oscillations. This is sort of, I gotta be honest, I don't understand these fully because I don't know what KD2 is yet. I don't know. But I'm gonna move on. So my goal, okay, that's really annoying. So I wanted to alter these models and sort of add on to them by adding parameters and changing equations, combining models, trying to see if I can find anything new and make it a digestible format, so anyone can start reading my notebook, and anyone with any background at all can start reading my notebook and then take away a thing or two. And yeah, thank you. I don't know if that was quick, but
That's great. That was great, and that's neat. You want to go back for a slide or two? I think this is a great choice for a whole variety of reasons. So go forward 1 slide.
yeah.
So what you have for KD2 is a parameter.
Okay.
Last week we just began to introduce the idea. I mentioned the word bifurcation.
Yes, it's
right.
been.
And we scanned how the steady states changed as we changed a parameter.
Oh.
So that's what this is. The steady states of P53.
Oh yeah, I read this. They were plotting the bifurcation what point? Right?
Yeah.
Oh.
In this case, those top and bottom dotted lines, P53 is not steady state, it's oscillating. P53 is going from a low value to a high value and then back to a low value again.
Yeah.
That's exactly what those high and low values are. You see that where it's got a picture of the oscillations.
You're talking about over here.
Yeah.
Or here.
No, on the right one. See, it's
Yeah.
showing the bottom line is the low value of P53 during oscillation. The top value is the high value of P53.
Got it.
We're not going to get to it today because we've been spending time on other things. But actually, one of the exercises that I had ready for this class, which we'll do next week, was in fact doing an oscillator. And we weren't going to do exactly that diagram, but I could show you how to do that diagram. Actually, I created a Colab notebook that would do that for you. Right. We haven't talked a lot about dynamical systems. If Connor were here, he'd give us code for this. There's some notation about how you plot what are called stable steady states, which are the solid black lines there. In that plot we were just looking at. The one under, if you move your cursor down a little bit, The solid black lines in the, yeah, there are the in the one that's the one to the middle, the one to the that one, yes, the one where you are.
Okay.
Oh, the one that says KD2 on the bottom.
Okay.
Okay, here what it says is to the left, but KD2 is small. There's
Yeah.
a single value of P53, and that's stable. If we push it up or down from that, it comes back.
Okay.
That's what the solid line means.
Okay.
If you keep going to the right a little bit, just a little bit,
Oh.
dashed line, and that means that if I'm exactly at that value, it stays put. But if I move off of it a little bit, it's unstable. I move away from it.
Oh.
That it's an oscillator. So what happens is, for a small, when KD2 is small, suppose KD2 is increasing slowly in time for some reason.
Mm.
P53 stays low
Mm.
and steady. Then it'll start to oscillate, and those oscillations get bigger. And if I keep pushing KD2 up, then I'll go back to a situation where I don't oscillate, where I have a single stable steady state.
Mm.
And so this is dynamical systems. There's a little bit of work that has to be done mathematically to understand how this works. Herbert has a chapter called Stability, and there's an appendix to the textbook where he discusses this. It's chapter 13.
Okay.
And there are other textbooks that I could show you that would cover this. But this is actually a really nice example. And I think what's happening here is that background in the ones which show time,
Yeah.
where there's that steadily decreasing function, there's that sawtooth, there's a triangle, in that sort of light gray triangle, that's something that's being imposed on the system. You cause damage at time 50,
Yeah.
and the damage is gradually going down.
Okay.
Changing one of these parameters. That's probably changing KD2.
Oh.
It's being forced slowly from outside the system.
Okay.
You're looking at how it responds. So that's actually, there's a lot. If you could duplicate those figures and have the user be able to change the time of damage, the amplitude of damage, they could learn a lot from this.
Oh, okay. Oh, wow. Okay. Thank
Now,
you.
if you go back to the previous slide, I just want to point out the model, as you say, is pretty simple. But one thing to learn, you don't, not now, but when you're going to be presenting a model, is that looking at those equations, 4, 5, 6, if I asked you, what does the first term in this equation mean? You should be able to answer that by the time you're done with the semester.
Okay.
And let's just look at equation 4 for the moment.
Yeah.
And let's walk through this, because this is what we did last week. And people were finding this a little bit confusing, I think, in the homework, which is, remember, we were talking about how to translate arrow diagrams into rate equations using Hill functions.
Mm-hmm.
And we said that when we have a gene regulatory network, we'll assume that there's a rate of basal transcription, which we call B, which is a constant. Look at the first thing there. What's the first thing after dp53 dt equals? What is it?
It's a constant.
It's a constant. That's the basal rate of transcription of p53. Then we said that the rate of production will be a Hill function of some activator. In this case, the activator is MDM2. Look what we have. We have a Hill function,
Huh.
right?
Yeah.
MDM2 to the 4th over constant to the 4th plus MDM2 to the 4th. That's exactly an activating Hill function of MDM2.
Huh.
And JZ53 is our K, our half maximal K that we had before. So the letters have changed, but it's exactly what we did in class last week. And then what did I say? Whenever you create something with a gene regulatory network, you have to have decay. What's the next term? Minus KD53 times the concentration of P53. This is a decay term. And so they've done exactly what we did last week for our example of autocatalysis. That's exactly how they built this model. They drew a diagram that says MDM2 promotes P53. And they wrote the simplest possible model that would do that. Does that help at all?
Yeah. Yeah.
Now, if we look at line 5, we have exactly the same thing, but now P53 is promoting the production of MDM2. We have basal rate of production, first term, the constant. We have P53 producing MDM2 as a Hill function, just like the previous line. We have decay of MDM2. But now we have one other thing that's happening, which we have not talked about, which we would normally talk about in the context of pharmacokinetic models. And this comes back to actually what I think Ibrahim asked about, what are compartments? So in your model, you have what's happening in the nucleus and what's happening in the cytoplasm. Right? You have MDM2 cytoplasmic, the MDM2 in the cytoplasm, and you have MDM2 in the nucleus. Two different regions, same molecule. And in this case, we've named the concentration of the chemical in the cytoplasm and the concentration in the nucleus as different species. One is 5 and one is 6. And you can ask, what is the rate of flow between the nucleus and the cytoplasm of the chemical? Well, the one that has the higher, the flow will be from the one that has the higher concentration to the one that has the lower concentration. Chemicals flow downhill just the way water flows downhill. And so those last two terms in the equations 5 and 6 are taking the difference in concentration between the cytoplasm and the nucleus and saying if there's more chemical in the cytoplasm than the nucleus, there's flow of chemical from the cytoplasm to the nucleus. If there's more concentration in the nucleus than the cytoplasm, there's flow from the cytoplasm, from the nucleus to the cytoplasm. If you'll notice, if you add the two last terms together between lines 5 and 6, they cancel. Because what those last two represent is the flow of chemical from the nucleus to the cytoplasm or vice versa. So those are called flux balance terms. If we only have one compartment, we don't have a flux balance. But in this case, with two compartments, we have to know how is chemical moved between the two compartments. That's what that's doing. Pedro, did you have a question or a comment?
Yes. Why would the decay constant be different?
Those aren't decay constants. Those are transport coefficients.
But in the last term.
You mean, why is it KD2 prime and KD2 are not the same?
Yes.
So the answer is that they're talking, because when you're doing transport of chemicals, you're not transporting concentrations, you're transporting amounts. Right? You're not when you have flux, you have amounts, not concentration. And so I have to convert the concentrations to the flux depends on the difference in concentration. Which way the flow goes depends from higher concentration to lower concentration. But I need to convert that to the amount of material that is transported. And then since everything is written in terms of concentration, I have to turn it back into concentration. And so I have to do a bunch of multiplication of the volumes of my compartments. So those KD, the reason those Ks have extra terms and have parenthesis marks on them is because the nucleus and the cytoplasm don't have the same volume. And this is correcting for the volume difference.
Nice.
I have a question.
Yeah.
Does this model take into account cell membrane that you have their different exchange, right? You have sometimes active channels that pump one concentration into the nucleus or out of the nucleus or so. This model is very simple. It's just a full permeable membrane in this case, right?
Well, it's semi-permeable membrane.
Or
because-
semi-permeable, yeah.
And I should be letting William answer these questions probably. And I've never read this paper, so I could be wrong. But yes, my assumption is that I think they're assuming there are pores in the nuclear complex, probably. If you go back to the the basic picture at the beginning. Yeah, the next one. Yeah, you see there are pores in the nuclear membrane. So you have transport food. They're basically holes in the membrane. So it's a semi-permeable membrane. So the reason to explain this, again, I could let Elmar explain it, but the reason this question is being asked is that if you have a membrane, you have some movement of chemical across the membrane, even if the membrane is intact. But often you have what are called channels, which are small holes in the membrane. And those channels have different ability to allow molecules through. Sometimes they have directional bias, so they'll let a molecule flow through more easily in one direction than the other, in which case you don't get an equilibrium. They're working effectively as pumps, so you don't have the flow stop when there's the same amount, this concentration is the same on either side. So you have ion channels, which selectively allow particular chemical species through. You have ion pumps that will tend to push particular species to one side or the other. And you can have holes that simply allow the molecules to flow through at a controlled rate. So all of these are possible. In this case, the assumption is that there's just a small hole and that the chemical flows across the hole at a rate proportional to the difference in concentration. But for other chemical species, if I were modeling, for example, if I were modeling the membrane of a neuron, where I have pumps that are actively pumping calcium, sodium, potassium, then I would have to have actually a model of how the transport across the membrane worked. If I look at something like Hodgkin-Huxley equation, the famous equation for how neurons fire, that's all detailed models of the ion channels in the membrane of the neuron, which are directional and gated. So the voltage across the membrane changes the flux. In that case, that K term would be a function of the values in the model. Okay, great comment, Elmar. Any other comments or questions about this? This is great. So the model looks very simple, but there's a lot there that you can do with it.
Yeah. I mean, well, they had four different models. So I just picked the first one. I think they were talking about positive. This was a positive feedback or something, and then another one was negative. I'm not sure. I'm not sure, but I guess
right. In this case, right, the more MDM you have, the more P53 you get. The more P53 you get, the more MDM2 you get.
Yeah.
Right. The Hill functions are both activating.
Yeah.
You could have a model in which one of those Hill functions were inhibiting.
Got it.
To do that, you'd replace the MDM2 in the numerator with the JK53 from the denominator. That flips the Hill function from being activating to inhibiting.
Okay.
So yeah, you could build a lot of models based on this. And of course, P53 is critical because one of the things that most cancers are highly diverse, but one of the things that's almost universal in solid tumors is that P53 is disabled. That's the single gene that's turned off in almost all cancers. And so messing up P53 behavior is one of the few really common ways that things that happen in cancer progression. There are a lot of other things that happen sometimes, but P53 is knocked out in a lot of situations. If P53 is overactive, you can have situations where cells die when they shouldn't die. Because the problem always in biology, this is true of the immune system as well, is that if something is going to go haywire, you'd rather have it go haywire by either stopping or dying, not by going berserk. Right? If you have a problem with a train, you want the train to stop. You don't want it to accelerate and run off the end of the rails. With an airplane, it's a little more complicated because you don't want the engines of the airplane to turn off automatically when there's a problem in flight. So a lot of biological systems are designed is the wrong word, have evolved so that they fail safe. Cells either die through apoptosis, which is P53, or they become senescent, they stop proliferating. What you don't want is the cell to proliferate improperly. On the other hand, damage happens all the time. There's always some damage to DNA as we age. There are cosmic rays, there's radiation in the environment, there's radioactive oxygen species damage. And if cells died every time there was any DNA damage, all of the cells in your body would die and you'd shrivel up. And so there has to be some balance between, gee, this damage is severe enough that I can't fix it, and I think that the best thing is for my cell to die, and gee, this damage is if I overread the damage and die when I shouldn't, then I'll have a degenerative disease. If I underread the damage and don't die when I should, I'll have cancer. And so making the choice about, is this level of damage one where I repair the cell, or a level of damage where the cell has to die because it's irreparable, that's a tricky thing to do. And so there are huge numbers of feedback loops, all circling around P53, not all, but many circling around P53, that try to determine, I can't expect to never have any damage. There's always going to be some damage to cells. Continually, there's damage to DNA, all the time. Most of that damage is repaired without problem. But occasionally, the cell will decide to repair when it can't. And that's one of the ways you get the initiation of tumors and eventually of cancer. And so this question about repair or die is actually a really critical one for cells to make. It's one of the most important cell decisions cells make. In some ideal world, we could say, well, you prevent damage to the cells, but there's always damage. There's always damage occurring. Okay. So I think this is a great educational model, and one can do a lot with it.
It's about to study transcription factor network of hierarchical differentiation of myeloid progenitor cells. And so first about biological background. So this is about blood cell differentiation in general. So you have first the hematopoietic stem cells, and they are actually located in the bone marrow, so inside the bone. And then they can differentiate in a big picture on two sides. So there are the myeloid sides and there are the lymphoid sides. In the myeloid sides, the development goes on in the bone marrow. And by the lymphoid side, they go into the lymph system, and that's part of our immune system, and they are developing there further. And this paper that I looked at, it only looks actually at the myeloid side. And then in the myeloid side, the cells can further develop to different types. So one of them is platelets, that are the cells that are, when you cut yourself, then those are the cells who fix the wound again and made new color chain and looked at that the tissue grows together again. Then you have the erythrocytes, which are the red blood cells. These are the ones that transport blood through our blood system. And then you have granulocytes and monocytes. So the I start with the monocytes. The monocytes are actually part of the immune system, and they can, when they invade, then they become macrophages. And macrophages are the cells that more or less, when we have an infection, so they are cleaning out the bacteria when they're dead, or just directly attack them and swallow them. So this is it's a huge part of our immune system. And then you have as well the dendritic cells. These are the ones who actually, when we generate an immune response, the dendritic cells are involved in getting B cells that are specific and have a B cell maturation that then in the end is specific to the virus that we attack. And then there are as well T cells. So the T cells, they are the ones as well attack the viruses. But I don't want to actually talk too much about the immune system. We stay here really on the myeloidic side. So interesting is just that the immune system itself, it has two sides. So one of them is actually myeloid. So it grows in the bone marrow, and the other one is really a lymphoid. And then you have as well granulocytes. They are as well part of the immune system. And so they are here, just all of these three cells are granulocytes, and they have actually, when you measure the pH of this cell, they have different pH, and that's how you distinguish them. So the basophils have more a basal pH, then you have the neutrophils, which are pH 7, and then the eosinophils, they are more acidic. So they stain differently when you have, for example, H&E staining. So this is just general about how our blood differentiates. Now the network model that I am looking at is really focusing on this breaking point that is here. This from the paper is highlighted, not in full color and not a little bit grayed out. And I translated those branching points into this big picture here where this happens. So you have the common myeloid progenitor cells, and they will differentiate in these megakaryocytes. They are mentioned here, and those are the ones that later on would develop into platelets. And platelets are actually a very interesting cell type. They have no cell nucleus, but that's another story. Sorry. And then you have the erythrocytes, which are the red blood cells. So they are called actually the same. And then you have this huge, then you have this GMP. So this is the one that differentiates. So this is the breaking point, this myeloblast, and it differentiates here in monocytes and in granulocytes. They are all just treated together. So there's not a distinguish. They don't distinguish between those granulocytes type. And here you have as well a branching point, the MEP, and actually in this image that I found, this one doesn't exist so as such, but I have to look deeper into this to see why this GMP branching point is here in this image source missing, because this is very detailed image that I found about the blood cell differentiation. And so they have, I think there will be a reason. So I have to find the sources and look deeper into this. So from the modeling background, I'm very interested in this Boolean generatory network. And we had at one point a question about what is different from a Boolean generatory network to when you, for example, use Markov chains or a Markov model. And I try here quickly to explain what the idea from a Boolean generatory network is. So we look at this artificial network. We have 3 genes, and all of these 3 genes are regulating each other. And you can see that the gene 1 here is regulated. So this is gene 1, and it's regulated from gene 2 and gene 3. And this is called an AND gate. So you can see when gene 3 and gene 2 is off, 0, 0, then gene 1 is off too. And when gene 3 is on and gene 2 is off, then gene 1 is off as well. And when you have gene 2 on and gene 3 off, then 1 is still off, but only when both, when 3 and 2 is on, then gene 1 is on. And then the gene 2 is actually regulated with the OR gate, and this is a little bit different. So an OR gate will actually switch when either of the other genes is on. So when gene 1 and gene 3 is off, then gene 2 is as well off. But when now gene 3 is on and gene 1 is off, gene 2 will be on. Or when gene 1 is on and gene 3 is off, we are still on. And even when both of them are on, we are on. So an OR, like we use it usually in the language, is actually not the same as an OR in logical gates. What we use in the language as OR is usually either OR, and this is called exclusive OR in the logical gates, but that's not shown here. Here is a real logical OR. So as soon any one of those is on, then the gene that is switched is on too. And then we have the gene 3, and it's as well regulated by an OR gate. And when we then translate these 3 genes, let's say in the beginning all of them are off, then in the next transition they will still be off. But when, for example, gene 3 is on, then it switches to that gene 2 is on and the other 2 are off. It's called a transition table. And now we can go look what happens here. So when gene 2 is on, then gene 3 switches on and gene 2 switches off again. And now we could go and look up what happens here. So here we end up actually in a cycle. And so this specifies the whole network, this transition table. We can then take this description that we have here and can study how many different cycles we have and as such how many attractors. So we see here, for example, when we are off, then the whole system will always stay off. And this is just one attractor. And when we not have noise and when we not can perturb the system, then the system cannot anymore come out of this attractor and it will always just stay switched off. Now, the second one that we actually looked at, the second 2 lines, was exactly like this, that it was actually a periodic cycle, right? It always was switching from this pattern to this pattern to this pattern to this pattern. So now we have an attractor, a periodic attractor. That's just what you showed before, William. And then when we look at the other states, so we see that there are states that are unstable and they flow towards, first, if we start by gene setting 101, you flow in the setting 011, then you flow in the setting 111, and this is again a point attractor, and you always just stay in this attractor. So I hope it's a little bit more clear how Boolean gene regulation works. So what of course can happen is that now through mutation, one of these genes get damaged, and all of a sudden our network changes. And now in this network that we have here, we have now and or. That's how our genes are regulated from the same network. And you can see that the whole landscape changes. Now we have here a single state that is a point attractor, and you come not out of this state. But before, when you were in the 111 state, now all of a sudden, okay, it's as well a state that stays, but when before we flow into the state 111, now we all of a sudden flow into the state 000. This one's changed as well. So there are other genes that have now this periodic cycle. So that's the theoretical background of how gene regulatory networks work. So in this study that I try to do, I really try to take a published gene regulatory network over that is about myeloid cells. And then in the first, I will have very steep hill functions. And then I tried as well to relax them. And there was another paper written from Wittmann in 2009. And he made a generic model over what is called hill cubes. And that's how with hill cubes, he defined his gene regulatory switches, so the Boolean gates. And with these hill cubes, it's not just that he only relaxes the hill function and make the hill function less steep. He does as well other things in how he really translates a Boolean network into a continuous network. And I really would like to understand as well this paper in detail and how we come from a discrete to a continuous network model. And here you can see that this was the first figure A shows the Boolean regulatory network. And of course, you have only two states, off and on. And you can as well see that because of when we go back here, this is very clocked and right, it's a time step and all of the genes are changing and the time step and all of the genes are changing, and that's how the whole network works. And this is not how biology really works, because you have in biology, you have regulation that go in different time scales. So some of them are very fast and some of them are slow. And so you can end up with artificial periodicity that you have here, right? It is off, then it's on, then it's off, then it's on. And when you relax the whole model and you translate in continuous model, you see that we lose this, for example, periodicity as we see, we lose it here. We get something else. We get like a different state in the middle by 0.3 expression. So we, when we, in this paper, they claim that when you relax the Boolean model, you get actually more attractors, you get more possible attractors that the system can reach. And I would like then to study the model that I have, the Myeloid model as well, to see, do I reach all of a sudden more attractors, right? How would this translate into what is known about the differentiation, and or do I not in this case, because it's very, very well known and the model really captures the big attractors and they just become a little bit different shape, but we don't get more states. That's things that I would like to investigate and through that get a little bit more understanding about gene regulation. So this is now this model that is specified, and there are in total there are 11 transcription factors. These are the orange square ones, and you can see here as well the activation and the inhibitor interaction, and these are 28 interactions, and the whole network is actually written down already. This is a logical description from each of the gates that we see here. So, for example, GATA2 has here GATA minus 2 and then this is GATA minus 1 and FOC minus 1, and then this is all negotiated. So this is an AND gate and this is another AND gate, and we have the negotiating of PU1. So not it's just changing the 1 to a 0. So the whole network is already described here. So in this Boolean update rules. So the first step, the first step will be that I just take this Boolean generator network and I translate it into Tellurium software using this hill function, and more or less I implement this Boolean updates rule into Tellurium. That's the first aim that I have. And then in the second aim, I really want to investigate now the model that I have. We have we had this functional network motif that we touched first time last class, where we had the bifurcation, and they are more of this network of this network functions, these fundamental functions around in this network. And I really want to look at the network and see what I find, which regulation, which basic regulation motifs I find, and see what I can find out. about this dynamic network motifs. And and in the paper they have as well described some of these regulations, and I want to see if I can at least find the same as they had in the original paper, or if I even see something else that I not have mentioned in the paper, or if I not can see something that they found, of course. And then in ENG3, I really tried, as I said, to relax the model, and I will, of course, first just maybe smoothen the transition so I have a lower hill power, that I not have these steep switch-like curves. But I really want as well follow this Whitman paper and see how they come from this discrete model to the continuous model, and then see if I have these changes. So what will change in my in having this all of a sudden this continuous model, and what I'm really interested in, will I find more attractors, because I think that's will be interesting to see. I will see. I'm really interested to see what I see there. So these are the references that I had. So the Boolean network was described by Stuart Kaufman. That was a book from the 90s. And then we have the model itself that was described, the Myeloid model in 2011. And then we had this transition from continuous from Boolean to continuous model that was described from Whitman. And interestingly, and also this is as well the reason that I want to do this comes all out of the same lab. So this is all of the Theis lab, and they are very, very active in studying single cell data, and I want to understand more the background from how they solve their stuff with their software. Yeah, thank you.
Yeah, wait, can you go to the graph and
Wait a second.
or the blue and red one?
The
That's not-
graph. Yeah,
Yeah.
here.
Whatever that is.
Yeah.
Okay.
So
can-
blue is on, and blue is off, and red is on.
Okay, but
And this is all the genes that are on the network. And you see that over time, these are time steps, right?
yeah.
And they see that over the time steps that now this gene, let's say the first gene, CD45, it is off, and now it switches on and it stays on for the whole time.
So wait, maybe I just completely missed the point. Why is it a scale if it's Boolean? I missed
in A, it's Boolean, but in B, it's now continuous. So this is when they did the transition, and you can see that you have then their states, like this state here, SAP 70, right? SAP 70 is all of a sudden 0.3, so it reaches a new level. It's not totally off and it's not totally on. It reaches new level just because we relax the on-off.
Okay. Oh.
Specification of the model, right? Yeah.
Okay.
So that's interesting. You can like think about the landscape, and all of a sudden you have as well hills inside, and not only mountains and seas, right? So it's something in between as well. So because of the relaxation.
Okay.
Yeah.
All right, cool.
Okay. Any other question? Or?
I mean, so one thing that's maybe important is that the basic idea behind the Kaufman idea of Boolean networks is the idea of transcriptional delays, that you have a time now when the gene is turned on or off, and then it takes some amount of time before the product is turned on or off again. That's why you can have steps. When you move to a reaction kinetic formulation, you don't have those delays anymore. If you want to implement those delays, you have to put them in by hand. And so one of the things that happens is when you have delays, you tend to get oscillations. That's typical.
And that's what we see here.
When you eliminate those delays, instead you get an intermediate steady state, which you see. And so one question is, in reality, there are transcriptional and translational delays. And so do you accept those or get rid of them? I think the methods that you're talking about don't. I think that the methods you're talking about are more or less exactly what I showed you how to do in class last week. There may be
I
some.
will. I'm okay. Okay. So there are some network, and because you have feedback loops, you have a kind of a delay when you relax the model. That's what I remember from reading the paper.
Well, it's certainly possible when you have more than three or more components in the feedback loop. It's possible to get oscillations. So it's not impossible to get oscillations in this network. But typically, well, we'll see. I mean, actually, the exercise I was going to do, the exercise that I assigned in the homework that I was going to do in class today, again, I don't think we'll get to that today. One of those motifs was a relaxation oscillator. And that relaxation oscillator has the possibility of either oscillating or being a steady state depending on the initial condition. So precisely you have a situation like this where under certain circumstances you get oscillations, but the same network for the same parameters may not oscillate for different initial conditions. And so that's exactly the kind of thing that you're talking about here. Here you have a cycle that depends on whether or not you start in a particular state. And so, yes, you can have this happen also for reaction kinetic kill function models. And the example that I gave in the homework was one of those.
Yeah.
So it's not impossible to get oscillations in these situations.
Yeah. But what they claimed is just that this oscillation is more artificial than. So this is actually another model, right? This is not the same model. This is another model, but in this model that they have there, they said that this oscillation that they have here is artificial and just caused by the way how a Boolean regulatory network updates, and they got rid of this the way how they translate this display to the continuous model.
That was what I just said exactly.
Yeah.
That because of the delays that are built into the Boolean formalism, Boolean models, when you have feedback, tend to have oscillations. When you put them into a reaction kinetic formalism with continuously variable rates, many of those oscillations will disappear. As you point out, not always. If you have the right kind of feedback, you can do it. But for example, let's look at the very first. If you go back to your 4, your 4.
This 4. Yeah.
So your cycle C, which is an OR cycle, you have number 2 turned on. The next time you have 3 turned on. Then you have 2 turned on. Then you have 3 turned on again. That will not be an oscillator in a reaction kinetic model. So you get both 2 and 3 turned on halfway.
Yeah.
So that will not oscillate.
Yeah.
You can only get oscillations with an inhibition.
-3.
No, you can only get oscillations not only with, you have to have at least 3, but you also have to have inhibition. You'll never get oscillations with purely activating states, which is what you have. So the rules for when you get oscillations are different.
Yes. Yeah. Yep. But I mean, that's kind of what I plan, what I would to do the minimum that I would like to do with this model and understand. So yeah.
But I think what you're proposing is fairly straightforward. I think it should be possible to do that.
Yeah.
And I think
That's the idea.
quite interesting to do. The next thing I wanted to do here was to talk a little bit about thinking about models. If people have taken other courses from me, they'll have heard the same discussion. So for those of you who've heard a course from me before, I appreciate your indulgence when we cover this material again. In the case of Pedro, he's actually helped teach this material, so he could help correct what I say. But very often we want to be able to engineer biological networks. We want to be able to have them do the kinds of things that Elmar was talking about. We want say a network to turn on when a signal is between two levels and turn off if the signal is below or above those levels. So a band pass. Or we want a network to turn on and then turn off again. Or we want to switch from a stable low to a stable high state when there's a transient oscillator. And so those are kinds of questions that actually are quite interesting. And actually, this is something that is exactly where those kind of Boolean network models that Elmar brought up can be useful, because the Boolean models are easier to understand conceptually, and you can use circuits to design a Boolean model, and then as Elmar said, back it off into a reaction kinetic model. Sometimes you get the same result, sometimes you don't. And you could do all of these things. You could have a circuit that turns on and then turns off again in time, a circuit that turns on again and off again as you change levels, pulse generators, and so on. But as an example of this, and it's going to come into a slightly different set of questions now, here's our feed-forward network. P1 regulates P3 directly, and P1 regulates P2, which regulates P3. This is called a feed-forward, three-element feed-forward network. And very often what we will do in biology experimentally is we will conduct a differential measurement. We will ask the question, if I increase P1, does P3 increase or decrease? That's a reasonable question. Why do we ask questions that way? Well, the main reason we ask questions that way is in biology, measuring absolute levels of things is quite difficult. Knowing exactly when something happens is often hard. Knowing exactly how much of something or what concentration of something we get is hard. On the other hand, if we have a fluorescent molecule, we can ask the question, is the fluorescence level, the level of fluorescence may not tell us exactly how many molecules we have. On the other hand, if the level of fluorescence is getting brighter, then that probably means the number of molecules is increasing. And so it's much easier experimentally to measure the directions of change, or is the change faster or slower under certain circumstances, than it is to measure how much do we have, how fast are things changing. And so very often, biological systems are quantified. If they're really made quantitative, we often have measurements in terms of what are called fold changes. The amount of something doubles, the amount of something goes up by a factor of 10, the amount of something goes down by a factor of 10. The time for something to happen doubles. And so this idea of differential measurement is critical to biology. And it's a little bit alien to the way that physics and mathematics tend to operate. It's not that you can't define these things in these other contexts, but it's not as necessary, perhaps. In physics, we measure the pressure, we measure the temperature. And certainly we'll say if we increase this, something else will increase or decrease. But usually we're able to measure those things. And biology, differential measurements come about very much because it's hard to measure the exact values of things. And the other thing that comes about in biology is that there's a lot of variability. There's a lot of stochasticity, random fluctuations. The times at which things happen often differ. You're exposed to the influenza virus. Someone else is exposed to the influenza virus. You both get influenza, but one of you gets it after 3 days and one of you gets it after 5 days. And those times are often variable. And different individuals will often have slightly different reactions. I give you the same dose of a drug as somebody else. In both cases, it reduces the rate of cancer growth, but in one person, it reduces it more than the other. And so this kind of variability means that looking for directions of change is often a better way of aggregating information, or more, better is maybe the wrong word, a more convenient way to aggregate information than absolute numbers. So that's something to think about. And so when we're building models, and this has come up in both of the examples William and Elmar presented, we want to think about what are the kinds of questions that we can ask. And actually, both of these examples will be things that I'll touch on in this little discourse that I'll provide in the next few minutes. Ultimately, and you'll see this diagram a lot when you take classes with me, and you'll see it multiple times. What is a model? A model takes a model structure. What is a model structure? Well, in our world, a model structure is that network diagram, like that Boolean network that Elmar showed. There are nodes that have states, and there are connections that could be activating or inhibiting. That's the model structure. There also are parameters. Those parameters could be the initial values of those nodes, or they could also be the strength of coupling. How strong is the activation or the inhibition? That wouldn't show up in a Boolean model, but it would show up in the real system. In any case, the model has to predict something, or it's not doing anything. And typically what it'll predict is either steady state values or time series. And we have to have some way of comparing the output of the model to reality if we want to be able to make it useful. So for example, in those oscillations of P53, in principle, we could measure the P53 level in cells using a fluorescent reporter and seeing how those oscillations happen in a real cell after damage. And what we'd find is that different cells might respond differently. The phase of the oscillation might be different from cell to cell. The level of damage that leads to oscillations might be different from cell to cell. But in general, when we damage the cell enough, we got oscillations. That would be an observation. And so I want to talk a little bit about what kinds of metrics we could use to make meaningful observations about a system. Again, if we're doing pure mathematics, we could write down some time series output of our model that we could never measure in the real world. And as a mathematical question, that's interesting, but as a biological question or an engineering question, it's not very useful because we don't know how to apply that in the real world. So let's think about our networks. Well, again, in most of our network models, the parameters are the initial values of things, which Elmar talked about, and also William, and the rate constants. And William showed an example of a bifurcation diagram where one of those rate constants was changed, and you saw that the behavior of the system changed as you changed the rate constant. Bifurcation diagram. The model structure is the interaction diagram and possibly the rate laws, the structure of the rate laws, but not the parameter values. The output of a model we're used to, the output model is going to be a time series of the concentrations of all of the species in the model, or something derived from that. Could be the long time values, the steady state values, if there are any, but it's a time series. And that means that there are a limited number of things we can ask. Now, when we do agent-based models, where our outputs are movies with cells that are moving around and doing things, it's harder to do the analysis. But for network models, we have a fairly limited number of things we can ask. What's going to be useful will depend on the situation and what we're trying to understand. But the first thing we can ask are qualitative questions. These are categories of behavior. And we've seen these both in William's example and Elmar's example. Qualitative questions are useful because in general, we can answer them without making detailed measurements. Is the cell alive or dead? It's a qualitative question. Does the time series reach a steady state, or does it oscillate? In both William's case and Elmar's case, we had situations for initial conditions or parameter values where the system reached a steady state. For others, we had oscillations. In Elmar's case, moving from the Boolean model to the reaction kinetic model changed whether you had oscillations or not in the example that he showed. But there are not that many kind of time series that you can have. If you think about a single line, what can the line do? It can reach a steady state. That's possible. It can oscillate periodically. That's possible. It can oscillate in a chaotic way. We haven't really talked about chaos, but that's possible. Or it can blow up. It can go to infinity or minus infinity. Those are the only things that it can happen. There isn't anything else. And so if I'm looking at a simple time series of one variable, there are 4 possible qualitative responses. It can reach a steady state, it can oscillate periodically, it can be chaotic, or it can diverge. And in biology, divergence is death. If the concentration of a chemical species goes to infinity, the organism dies. So that doesn't seem like much, but you can ask more elaborate questions. For a given model with a given set of parameters, boundary conditions and initial conditions, which of these do I get? Are there different initial conditions for the same model that give me different states? For the same initial conditions, if I do a perturbation, can I go from one kind of state to another? Those are all meaningful. If my heart is oscillating, which it better be, and I get hit in the chest with a baseball, if my heart stops, I go from an oscillating to a steady state situation. That's not good. You can model that. If I get hit in the chest with a baseball and my heart goes from an oscillating to a chaotic system, which is fibrillation, that's also not good. That can be modeled. And so these transitions can be quite important. They can be literally life or death decisions. And please stop me if there are questions, because these seem to be very simple concepts, but applying them in your own work is not something that's trivial to do. Okay, so the key question then is, if we change something in the model, Elmar talked about a perturbation of the model structure. We turn a gene on all the time instead of having it controlled. We take a gene out of the system. What happens? William talked about a perturbation where we cause DNA damage. We increase the level of damage. We have a spike of DNA damage, and then the DNA damage decreases. What happens? Do we switch from steady state to oscillating, or oscillating to chaotic, or chaotic to divergent? These are critical questions. We may want to know how many different states of each type do we have. We could have 2 different oscillating states for the same parameter values. One could be a fast oscillator, one could be a slow oscillator, for example. What are the boundaries between the states? If you want the cell cycle, in cancer, we have cells proliferating improperly. Well, we might want the cell to die when we treat it with a cancer drug, but we might want the cell to stop dividing. We might want to knock the cell from an oscillating state to a steady state. If we want the cell to die, we might want the cell to be knocked from an oscillating state to a divergent state. Those might be important things to understand if we want to, say, use an antibiotic to kill bacteria without killing the host, or chemotherapeutic. to kill the cancer cell without killing the host. So these boundaries between different types of regimes can be very, very important, even if we can't quantify the exact behavior within the regime. So if I have a set of parameters that I could control, KD2, the coupling strength, the speed of reaction, all of these things, and I plot them. Now, in reality, there may be many parameters, and if there are a lot of parameters, it's not so easy to plot them all at once. So you tend to do cuts. You maybe do 3 at a time, and then you do cuts. But let's imagine that there are 2 parameters, and here, this is a fantasy. It's not the real world. But suppose that I'm imagining the heart, and it's oscillating. And I say that I change the extracellular potassium concentration as a parameter, and I change the extracellular sodium concentration as a parameter. And if I have a very low level of potassium and sodium, the cell doesn't oscillate. If I have a low level of potassium, no matter what the sodium concentration is, the cell doesn't oscillate. If I have a very low level of sodium, no matter what the potassium level is, the cell doesn't oscillate. Well, if it's a cardiomyocyte and the cell doesn't oscillate, I'm in trouble, but okay, this is a fantasy. Now, suppose I have a little bit more potassium and sodium. There's a regime where I have a moderate amount of both, where I get oscillations, where the cell behaves properly for a cardiomyocyte. Now, I have a potassium level that's giving me oscillations. I somehow increase the amount of sodium in my environment too much, and I go from an oscillating to a chaotic state. I start having fibrillation. Well, that's not good. So this represents too much sodium in the system is causing a pathological change in the behavior. Similarly, same thing happens. I'm in a good state. I put in too much potassium. I get a chaotic system. And in this case, I've drawn something where if I keep increasing the potassium level, it's toxic. The system diverges and eventually the cell dies. So this little diagram is telling me that if I want my cell to stay alive and when I want my heart to work, the potassium and the sodium have to stay in a given region. If I go too high, I get a particular kind of failure. If I get too low, I get a particular kind of failure. Now, this is very much a fantasy. I'm not saying that this particular diagram is real, but you could see perhaps how you could use a diagram like this to design an intervention to treat a heart disease or to predict a kind of heart disease. And so if you can build these kinds of structural relationships, then you've learned a lot about how the biology of your system works and how to control it. In general, the exact positions of these lines in terms of sodium and potassium are not going to be right in your models. However, if you built your model structure properly, the identity of the region, steady state, oscillating, chaotic, divergent, and the fact that there's in a particular sequence, those things should be reproduced properly by your models. It's reasonable to ask that that happen. If they're not, then your model has a limited domain of applicability, and you need to know that because that's a limitation. So this is what's called a phase diagram. Now, physicists use the term phase diagram. We have, if the temperature and pressure are this much, water is ice. If the temperature and pressure are this much, we have steam. If the temperature and pressure are this much, we have liquid water. And in the case of ice in particular, there are many, many different kinds of ice. All of them are solid, but they have different structures if we change the temperature and the pressure. And so in that case, we'd have very high temperature and low pressure, we have steam. Intermediate temperature and pressure, we have water. Low temperature and high pressure, we get ice, and we get all sorts of different ices and transitions between them. So that would be a phase diagram for a non-living material. But these phase diagrams for living materials are one of the key things that we do with the kinds of models that we build in this class.
Can I ask a question to that?
Please.
I mean, when you now just have solid, liquid, and what was it? And gasoline form. No, gasoline is wrong.
Yeah.
Gas form and solid and liquid, but there you don't have a. Or what is now your divergent and your chaotic state? I mean, I see that you have different regimes, but I cannot now take the states and put it into this 4 that you described and said it's the only one.
No, but for materials, the states are different. I'm only talking about a time series of a scalar value of a chemical concentration.
So the 4 ones that we defined are for time series from chemical concentration.
That's right. Yes.
I see. Okay. Now that makes me not clear. Thank you.
Right. There could be, I mean, depending on the system we're looking at, there might be many states. If I'm driving a car, one of the states of the car could be has flat tires. One of the states of the car could be sliding on the road. One of the states of the car could be crashed. There are many possible states in the real world. But if I have a time series and the only thing I have is a value at every time, there's a limited amount of things that can happen.
Yeah, thank you.
An additional thing is that for a biological time series, concentrations change continuously. They can't jump discontinuously. And so I'm also assuming that there's only continuous change of value. And otherwise, there are more possibilities. So absolutely, if I'm not dealing with chemical concentrations, there are more things that can happen in the real. That's why I was saying if I have a model of, say, cells in a tissue, and the cells are able to move around and reorganize, that allows me to have lots of state possibilities that I'm not talking about here. But in the case of the network models we're dealing with here, the number of things that we can actually have qualitatively is quite few. There's still plenty. There's still plenty of richness, but it's easier because it's not so much to interpret. Okay.
Thank you.
Great. So those are qualitative questions. We don't have to know numerical values of the outputs. We just watch the shape. We then have the classic biologist question, which is a semi-quantitative question. Suppose we're using a fluorescent label for a chemical. We don't know what the fluorescence intensity corresponds to an exact number of molecular concentration. This intensity is this many moles of the chemical. We don't get that. Depends on our microscope. It depends on the chemical we use. It depends on the way we light it, all of these things. It depends on the size of the sample. On the other hand, as I mentioned, we can often detect trends. The fluorescence is increasing. The fluorescence is decreasing. So the classic semi-quantitative question is, if we do something, does the level increase or decrease? As time goes on, does the value increase or decrease? Even if we don't know the absolute value, we know whether it's going up or down. That's very important. The time example of that would be the order in which things happen. Does A happen before B or after B? Not when does A happen and when does B happen, but what's the order? That's often easier to observe. We don't need to make an exact measurement, but we know that the cell goes through the myeloid precursor state before it's a myeloid cell in that differentiation pathway, for example. And very often we'll ask a question of a stimulus and a response. If we increase the level of molecule A, does the level of steady state of molecule B increase or decrease? If we knock out gene X, does the level of a molecule increase or decrease? Does increase of molecule A increase or decrease B? That is A an activator or an inhibitor of B? These are the kinds of classic semi-quantitative questions. They talk about directions of things and relationships between directions. It can be a little bit more complicated. if A is increasing, does the rate of increase of A increase or decrease? Actually, I have it backwards here in this one. If B is increasing, does increasing A speed up the rate of increase or decrease the rate of increase? So does A accelerate or decelerate the production of B? That's also a semi-quantitative question. Okay. These kinds of questions can be pretty important, and the boundaries between these things are also important. If A is at long times, is A bounded? Does A go to 0 or does it go to infinity? Well, bounded versus infinity is a qualitative question because we distinguish steady state from divergent. But 0 value bounded versus finite value bounded is now a semi-quantitative question because it's relative levels that we're talking about. We can build phase diagrams for that too. Suppose we have two chemotherapies, one of which is a classic chemotherapy, and one of which is an anti-angiogenic chemotherapy. And we say that if the chemotherapy level is very high, the tumor shrinks. If the anti-angiogenic level is, well, I've drawn something, again, this is a fantasy, so I don't know how realistic it is. But certainly if I give no chemotherapy and no anti-angiogenic, the cancer grows. If I give a lot of chemotherapy, probably the cancer will stop growing. So I can draw the boundary line between tumor grows and tumor shrinks on my control diagram. Now, one of the things that I know is that if I give too much of a drug, it's going to kill normal cells. And so here, if I give a low dose of chemotherapy and a low dose of anti-angiogenic, my cells are okay. My regular cells, my normal cells are okay. If I give too much of it, my normal cells die. Well, okay, those are both separate observations. Now I put them on top of each other, and now I have a green region, I hope, if I'm lucky, where giving that chemotherapy and that anti-angiogenic therapy, the cancer cells shrink, the tumor shrinks, but the normal cells don't die. And that green region then would be the therapeutic region for these drugs. If I was unlucky, no matter those would be non-overlapping. If anything where the tumor shrinks, I kill all my normal cells. Then there would be no way of getting a therapy out of this combination of medications. So these semi-quantitative questions can be pretty important. They can determine whether a therapy works or not. Now, we might not know the exact values because our model might not give us the exact values that we need. But if I look at my picture here, what it's telling me is that there's a narrow band of chemotherapy concentrations which might work. And if I give too much anti-angiogenic, it's never going to work. And so even if I don't know the exact values, it might give me guidance about what kinds of combinations to try in an experiment or clinical trial that are most likely to work. And then I can calibrate from them all. Okay. And the last kind of questions are quantitative questions, where we actually measure things. What is the level of something? That typically is very hard to do for chemical species. It may not be as hard to do if we're measuring something like how much does something weigh? How big is the tumor? Although actually measuring a tumor in a live individual is not so easy. How fast is something increasing? When did something happen? How often does something happen? That last one may be a little bit easier. How frequently does something happen? May be easier than exactly when. So those are quantitative questions. But there's a hierarchy that qualitative questions are easiest to measure in experiment. Semi-quantitative questions are what biologists typically measure in experiment. They're a little harder to get than the qualitative ones, but they're usually possible. If I absolutely have to know quantitative results, it typically is pretty hard. And so if I'm going to compare a simulation result to an experiment, the easiest one to compare would be the qualitative one. The second easiest would be the semi-quantitative, and most validation studies of simulations are done with semi-quantitative methods. And the hardest one is the quantitative one, because generally we can't make those quantitative measurements in an experiment. These are more quantitative questions. Is the concentration of A more or less than B? That may seem semi-quantitative because it's a comparison, but I can't make that distinction. I can't make that comparison just by measuring A's change or B. I have to know both, the values. Is the rate of increase of A greater than or that of B? We can't do that without knowing the exact values. What's the exact functional relationship between A and B? These are all things that are pretty hard to measure. They're important. We'd like to know how much of the drug gets into the tumor. Well, maybe that one we can do with a radioactive tracer. How much ethanol do we produce per unit of glucose? But those are also very susceptible to the details that we leave out of our models. And so in general, we can't really expect that we're going to get quantitative predictions exactly right for the kinds of models we do here. Now, if I'm doing drug dosing and I'm doing pharmacokinetics, and I take a pill and I know how many grams of drug are in the pill, and I want to know how much of that drug gets into the tumor, well, in that case, I have to do the hard work to get the quantitative results. But there's going to be a big uncertainty band on it, even with all that hard work. And the value of a company like Certara that does pharmacokinetic models is they've done decades of calibration to make sure that those quantitative results are right. In this class, of course, we're not doing that. But if you were taking a pharmacokinetics class, a lot of the class would be about that kind of calibration. You could spend actually 2 semesters doing nothing but understanding how to do that quantitative calibration in that specific case. It's not easy. I was going to ask you to think about it. And as I say, in a sense that the order here was very nice, because both William and Elmar showed examples of all this kind of thinking in their cases, in their examples. And so you could maybe go back and using this formalism of what are the boundaries between different kinds of behavior, quantitative, semi-quantitative, qualitative. What are reasonable questions to ask about your model system? As an example, in William's case, it could be, under what situations of damage does P53 oscillate or not? Does P53 blow up, causing the cell to die or not? Those would be reasonable questions. What are the things that determine whether that happens? As an example, there probably are a lot more. For Elmar, a question could be, what are the factors that determine the ratio of cells that differentiate in one pathway or another? If you perturb a particular chemical, which represents a parameter in that model, do I change the ratio of the cell types? That would be a kind of question you could ask. But I'd like you to pick 2 examples. One could be your project question, if you're ready for that. Another one could be something else, but I want them to be different so that you have to think about it a little. First what are the variables that you're going to measure? How are you going to quantify the system? You're going to measure the level of P53 and something else, maybe? You're going to measure the fraction of cells that become myeloid or the level of a particular protein. What are the variables you're going to consider? Can they be measured? Very important always, if we want to come back to experiment. Is it even possible to measure them in an experiment? And if so, what are the limitations of those measurements? Again, I'm not asking you to do this in enormous detail, but if you're predicting something that could never be measured, even in principle, then that's not as useful as it is to predict something that you could measure. And then, what is the behavior you wish to determine? What is it that's important? Does the cell live or die? Do you get are you missing a cell type during differentiation? Do you get too much of a cell type? Do you get an uncontrolled number of cells and get a leukemia? Do you get an uncontrolled number of erythrocytes? And I can never remember the name of the red blood cell cancer that is much rarer than leukemia, but can kill people. It's untreatable at the moment. And then why is the behavior significant? And so for your problems, ask each one of these things. What's a qualitative question you could ask that's important? What's a semi-quantitative question you could ask that's important, and why? What's a quantitative question you could ask, and why? Does that make sense to people at all? Is that a reasonable way of framing this? I know it seems maybe a little bit abstract, but I was hoping that after what Elmar and William showed, those are exactly the kind of questions that those papers you're looking at are trying to address. And when Elmar asked the question, if you go from a Boolean model to a reaction kinetic model, how does the prediction of the qualitative behavior change, right? You went from a Boolean model to a continuum model, and you went from having oscillations to not having oscillations. But that was an aspect of the model. It wasn't an aspect of the underlying network. And that means that the way you were doing the calculation was messing things up, right? It was giving you the wrong prediction. And so that's an important thing to know, very important thing to think about. Because we know that the ways we're doing these calculations are approximations. For gene regulatory networks, genes are typically on or off, but then the levels of proteins change in particular ways. There's stochasticity we're leaving out. There's spatial variability we're leaving out. And so we'd like to know, are predictions sensitive to the details of the model that we're leaving out? Are the assumptions we're making giving us the wrong answer, and how? And so I think that actually the two examples that we saw today are wonderful for illustrating why it's important to think about these things. And I understand it's not easy to ask these questions and answer them, but I think it's worth challenging yourself a bit and exploring these ideas. And primarily, of course, I'm asking you to do this for the purpose of your project. And if you're going to go on and do research, whether you're doing experimental research or computational research, modeling, these are the kinds of questions that you have to ask. It's distressing that people build computational models. They run simulations and they get some output. And they say, we got some output. But there are a surprising number of papers which never ask the question, how do we extract meaning from this output? Why is this result significant? How do we use it? How do we compare it to reality? How do we apply it? And I think it's worth asking from the beginning when you're designing a question and designing a model, how are we going to apply this in the real world? Why will it be meaningful? What is its sensitivity? What is its range of validity? If we're making approximations, are those reasonable or not? And so I think at some level these are very fundamental, and they're not easy. It's easy to write a question like this. To answer it is not so easy. And but I think grappling with it is important. And again, if you're doing engineering, you're going to need to grapple with this. Whether you become a bioengineer, a mechanical engineer, an electrical engineer. Electrical engineers mostly deal with quantitative questions because they can measure things more easily. But you have to address these questions in every field of engineering, in every field of science. And network modeling is nice, and biology is nice because it forces you to think about these things from the beginning. You can't take any of these things for granted. You can't take the ability to measure for granted. You can't take the ability to compare for granted. And so I think that this is, in a sense, a great set of training exercises that will be helpful no matter what you do.
No, I had a question. You said some of like papers are being published and they don't draw conclusions from what they get, right? Like you were talking about how the mathematical models, and they just get some make some conclusion, but they don't ask the correct questions and answer the correct questions. How are they getting published in journals then?
There's a field of mathematical biology. Mathematical biology is a funny field because it's between biology and mathematics. And there are two very different philosophies of mathematical biology. There's what a biologist would do or what an engineer would do, which is to say, here is a biological problem. Can we use mathematics and computing to solve this biological problem? And that's what we're talking about here. On the other hand, if you're a mathematician, really a mathematician, you might say, here's a biological problem. Here's the model structure of this biological problem. It suggests new and beautiful mathematics that is beautiful as mathematics. And the fact that it's mathematically beautiful may not have any relevance at all to biology. It's basically mathematics inspired by biology. And so, for example, we have people in our mathematics department who say there are particular differential equations that are inspired by a biological problem. And they ask the question, are these differential equations formally soluble? Can we prove that they have solutions under particular conditions? That's very beautiful mathematics, and maybe in the long term, those proofs come back and are useful. But fundamentally, they're not asking the question, is this useful biologically or from an engineering point of view? They're asking, have we gotten some new wonderful mathematics that we can that is inspired by a biological problem?
Got it. Okay.
And so in that case, there may not be something that's directly measurable in the real world, but nevertheless, it's quite beautiful mathematics. I mean, I'd be the last person to say that pure mathematics can't be useful, but people don't do it to be useful. They do it because it's beautiful. And so, I mean, if I were giving a course called mathematical biology, I would spend more time on the beauty, perhaps, and less time thinking about how do we use these things in the real world. This course sits in an engineering department. And as engineers, ultimately, the things that we do may not be products in themselves, but they need to be connected to the possibility of making a difference in the real world. And so I'm focusing on ways that we can connect the mathematics that we're doing and the modeling we're doing to real world questions. Do I think that everything we do has to have a real world result immediately? No. We can do things that are a step behind it or 2 steps behind it. But nevertheless, looking over our shoulder, we should always be thinking, what are the possible applications? Whereas a pure mathematician might say, this is beautiful. I don't care whether there are any real world applications. And that's a legitimate thing to do. But then in a sense, you're an artist. And we need artists. But it's a different set of questions. The most enjoyable thing, of course, is when it's both beautiful mathematically and it's useful. But things could be useful but ugly. I would say flux balance analysis is useful, but it's not pretty. It's not pretty. A lot of engineering is about solving problems. You don't care whether the solutions are beautiful. You care about whether they work. There are a lot of kludges in the world that work. Similarly, on the other side, it could be beautiful aesthetically and intellectually, but not useful, at least not at the moment. Maybe it comes together later. I'm not trying to make a value judgment. Where I would make a value judgment if somebody writes a model down that's not very beautiful mathematically or interesting, and it could never be validated in the real world.